Starting up.
Hi everyone, thanks for coming today. My name is Sarah, and before I dive into my project, I'm going to share a little bit about me. So the seeds of my honours project were sown back in 2023 when I completed a summer internship at the CSIRO. This experience took me to Samoa, where I investigated the adoption of intercropping practices in taro farming systems. Thanks to the Crawford Fund and with the support from my CSIRO supervisor Dio, I was able to continue this experience in the international ag world for my honours project. After much discussion and planning, we eventually settled on the topic, the adoption of agricultural mechanisation in Fiji. So let's get into it. Agricultural mechanisation is the use of machinery and technology to automate farm tasks, allowing farmers to operate with less labour in less time and at lower costs. In many cases, this has driven productivity gains and enabled agricultural expansion. But its impacts aren't all positive. Mechanisation can also lead to soil compaction, erosion, and rural depopulation. Additionally, while many countries have benefited greatly from mechanisation, in others, adoption has remained low, and this has been the case in Fiji. Fiji's agricultural sector is facing significant labour shortages. On-farm mechanisation in the form of small and heavy machinery is seen as a way of addressing this challenge. As such, the Ministry of Agriculture have made mechanisation a priority by introducing subsidies and loans to encourage adoption. But despite these efforts, adoption remains low, with only 14% of farming households using machinery. I wanted to understand why this was the case and set about to do so with the following research question. How do agricultural stakeholders in Fiji perceive the factors and interactions shaping the adoption of agricultural mechanisation? Because I investigated perceptions, I used a qualitative approach. Unlike quantitative methods that look at numbers, qualitative research focuses on perceptions, experiences, and behaviours of participants, answering how and why questions rather than questions of how much or how many. This study therefore sits in the interpretivist paradigm, whereby reality is assumed to be subjective and people shape their ideas about reality through their experiences. This means that my findings aren't presented as facts. Instead, they reflect how people perceive mechanisation in Fiji, which is valuable because people often act on what they believe to be true. Importantly, this paradigm acknowledges that the researcher plays a really important role in the research. My interpretation shapes how the research is understood. I used systems thinking as the framework for this study. Systems thinking takes a step back and looks at the whole picture. It helps us understand how different parts of a system interact rather than viewing them in isolation. In agriculture, this is really important because social, economic, environmental, and policy factors are all connected. So to answer my research question, I travelled to Fiji and performed semi-structured interviews with 10 key stakeholders. Through open-ended questions, I explored their experiences in agriculture, mechanisation trends, and its key drivers, barriers, and impacts. This conversational format encouraged participants to elaborate on topics they felt strongly about, giving me deeper insight into their perspectives. The first step of data analysis was developing cognitive maps for each participant. These maps visually represent a person's ideas and reasoning and help capture the complexity of how they think about a topic. On the screen are 2 examples of these, but don't worry, you don't need to read them. They're just there to give you a sense of what they look like. Next, I coded the interview transcripts using NVivo. I took an inductive approach, meaning the key themes and factors emerged naturally from the data rather than being defined beforehand. This allowed me to see what patterns and ideas consistently came up across the participants. I then combined the cognitive maps and the thematic coding results to create a single causal loop diagram. This diagram shows how different factors are connected and influence each other based on how participants describe them. This tool helped me identify feedback loops, sequences of events, and leverage points, which are places where a single change can have larger impacts across the system. Believe it or not, this is actually the simplified version of the map, and it took me many iterations to get it this clear. This map captures my key findings, which I've further broken down into smaller causal loop diagrams, and I'll walk you through some of those today. But first, a quick note on how to read them. Arrows come in two colors. Blue shows a positive polarity, meaning that both variables change in the same direction. When the first goes up, the second goes up. For example, as the shift to non-ag employment increases, labour shortages also increase. Orange shows a negative polarity, meaning when the first goes up, the second goes down. For example, when machine durability increases, machine downtime decreases. You'll also see loops labelled R and B, but I'll introduce those when we come across them. So that brings us to my key findings. Due to time constraints, I'll only be sharing the first 4 of these with you today. Labour shortages emerged as the most significant driver for mechanisation. These shortages stem from the perception that farming is unprofitable, strenuous, and high risk, prompting a societal shift to non-ag employment. This contributes to labour shortages, which reduce the profitability of farming, ultimately reinforcing the negative image of agriculture, making this a key reinforcing feedback loop, as indicated by the letter R, where each factor amplifies the next. Mechanisation is seen as one way to counteract this loop by reducing reliability on manual labour and improving farm profitability. However, participants cautioned that as mechanisation replaces manual labour, more workers are pushed away from agriculture, therefore worsening the very problem mechanisation aims to solve. For my second key finding, participants stated that the adoption of mechanisation was greatly influenced by farm-level characteristics. Having a small farm was described as a barrier, as they often lack the economies of scale required to justify investment. This barrier is worsening due to social change. As cities grow, peri-urban farmland is converted to residential lots, and this, compounded by land tenure insecurity, is reducing available farmland and farm size. Agriculture is then pushed to more marginal and mountainous areas where mechanisation is less viable. Fiji relies heavily on imported equipment, which is both expensive and takes a long time to arrive. As a result, prices stay high, demand stays low, and suppliers have little incentive to import more equipment, creating a reinforcing cycle of low availability and high costs. Another major issue is access to repair and servicing. Many suppliers don't stock spare parts, and there's a shortage of skilled technicians. So when machines break down, they remain out of service for long periods. These factors contribute to the perception that mechanisation is both unprofitable and high risk. In this image, we have Dio, my supervisor, standing next to a handheld rotary hoe that has clearly seen better days. My fourth key finding was about access to information, which includes how to use, maintain, and purchase machinery. In Fiji, it is restricted not just by the limited engagement from extension officers, but also by their capacity to share knowledge, as many of them aren't trained in machinery operation or maintenance. Training at the point of purchase is also minimal, So when farmers do mechanise, they often lack the skills required to use machines safely and sustainably. This leads to accidents and environmental degradation, which strengthen the perception of mechanisation as unprofitable and high risk. As a result, adoption stays low, demand for information remains weak, and access to information continues to be limited. From mapping the system, I identified a few key leverage points. Remember that leverage points aren't actions or interventions, they're places within the system where a single shift can create systemic change. The first is land tenure. Many farmers are uncertain whether their leases will be renewed, which makes them hesitant to invest in machinery or long-term improvements. By strengthening trust and confidence in the leasing system, farmers may be more willing to invest in agricultural development. Another leverage point is access to repair services. Because spare parts and trained technicians are scarce, machinery can sit idle for months, reducing reliability and discouraging further investment. Improving local repair capacity could break the cycle by keeping machines running and restoring farmer confidence. And finally, perceptions of agriculture. Farming in Fiji is seen as unprofitable and high risk. But if this mindset changed, it could spark greater youth interest, government support, and private investment, making the whole system more resilient. Shifting perceptions or mental models of agriculture has the greatest leverage in the system, but is also the most challenging to achieve. So the findings from my research show that mechanisation adoption in Fiji is a really complex issue, integrating social, economic, environmental and institutional factors. For R&D, it highlights that before we promote innovations, we first need to ask questions. Questions about the challenges faced by communities, what people want and value, how local systems operate, and what kinds of support are needed to make change last. By listening to those directly involved, we can co-design interventions that actually fit their realities. So that brings us to the end of my presentation. I want to say a really big thank you to the Crawford Fund for making this project possible, as well as my supervisors Dio, Deepa, Aaliyah, and Al for guiding me throughout this process, and my partner Hugh, my family, and my friends for being the best support. Thank you very much.
Thank you, Sarah, for that presentation. Time for questions. We'll have about 3 minutes. Could spare a little bit more, but yeah, officially 3 minutes for questions. Anyone?
Sarah, that was an excellent presentation. Thanks very much for sharing your experiences. It's really good. So you're saying the leases is a critical point. I may have missed the introduction, but who manages the leases and how long are the leases for? Out of curiosity.
So I'm pretty sure it's 65% of the land is held under traditional lease, traditional ownership. Only a small proportion is held by the government's leases. And I think they were maybe 20-year leases, but all of a sudden they've all come to the end. And yeah, so sorry, what was your question exactly?
That was basically this. If the lease is only 20 years old, 20 years,
yeah.
You can understand the the uncertainty and anxiety and not wanting to invest in the mechanisation if they're so short.
Yeah, exactly.
Yeah, so that makes sense. Yeah, thanks.
But the original question was who manages the lease? Did you say that?
Oh, sorry.
Who manages the leases?
Manages the lease.
Yeah. Are they government managed?
Yeah, the government and private
The mixture.
ownership as well. Yeah. So they have their own agreements with people, but I think the regulation around those agreements is pretty poor.
Do you have any feel for the split between government and private? Is it mostly private or mostly government?
I'm not certain.
Yes.
You said at the end, your last slide, that about designing interventions. What kind of interventions do you envisage?
Do I envisage? It's a tricky question. Well, from my research, I've indicated where interventions could be effective, but exactly what those interventions look like is a tricky thing to navigate. And I think you would have to have a lot of people in a room together representing lots of different perspectives coming together and figuring out something that works for everyone, but I'm not exactly sure what those interventions would be. I know that education is something that needs to be improved. As I said, land tenure, access to finance is a really big challenge in Fiji, especially for mechanisation. I had that on my here. That's one of the key leverage points, because if the farmers can't access finance, it doesn't matter if they have access to repair services or their land tenure is in good shape, because they can't even buy the machinery. So that's one of the key interventions I think would need to take place.
What about the role of extension? Are there extension people there that can help provide this, or is that something they could add in?
There is extension. I spoke to some extension officers, and I think the biggest issue that I found was that there's not enough of them. So like each extension officer will be managing hundreds of farmers, and they just can't visit all of them. One of the participants even said that farmers in the very inland or regional parts of Fiji sometimes have never been visited by an extension officer. But also they lack the skills required to share the information.
Thank you very much.
In terms of access to finance, was there any evidence of that you picked up of sort of farmer groups getting together to collaborate?
Yes, there is. I actually have a slide prepared. Access to finance.
Plants.
So farmer cooperatives are being used to help farmers access finance, but one of the biggest challenges with the farmer cooperatives is they don't have clear responsibilities or agreements. So when they get a loan or they purchase a machine, they don't decide, who's going to be responsible for maintaining it, who gets to use it first, and who's going to pay for this and that. And these, I think there is a quote on here, maybe not. But a lot of the cooperatives have failed. That was a big issue.
That's a question. I was interested when you said that mechanisation is driving people away from agriculture. Is it driving? Obviously, mechanisation replaces the need for some labour, obviously, but is it driving more people away than so that there's even a greater lack of labour following the implementation of mechanisation?
I can't speak to the exact numbers, but I can say that there were concerns raised about people being pushed away from agriculture.
Sure.
But some participants did also note that with mechanisation comes the requirement for more technical knowledge, which may actually attract more commercially mindset people into agriculture.
Sure.
This image, sorry, just back a little bit. This image actually shows that, so you can see, sorry if I'm taking up too much time, but so this loop here shows how mechanisation contributes to farm labour shortages, but down here, requirement for technical knowledge and commercial mindset people attracted to agriculture are actually reducing farm labour shortages.
Right. Have we run out of time for questions?
I just want.
I've got one more question, and that is, what are the learnings for Tasmania here as well? We're on the cusp of implementation of technologies and AI and automation and there's a whole bunch of stuff that's coming down the track towards us here in Tasmania. And is there any learnings for Tasmania here, do you think?
I think there are. I think that the way I've approached this problem could be used in many different contexts, such as Tasmania. And by getting lots of different stakeholders together, trying to really understand the system and what's happening and how these technologies will affect the system, I think that would be really useful. So, yeah.
Thank you, Sarah. Thanks everyone for your questions. Can we give a round? So let's welcome our second speaker, who is Rupert. Rupert is speaking on analyzing the effect of mandatory soil amendments on water in apple trees using stabilized carbon and nitrogen isotopes. Let's welcome.
Right, I can make this a bit bigger. So, big as we can. No notes. No notes. Okay, well, thank you everybody for coming, and thank you, Arpe. So let's jump straight into it. And how do you work this thing? I'm a Mac operator. Beautiful, perfect, thank you very much. So very quickly, we're going to set the scene and frame my project. You can see there in the top right-hand corner that Tasmanian horticulture is worth a lot to the agricultural sector in Tassie. And kind of the key prompts for my project are around the increase in climate variability. We've got the increase in the expense of irrigation generally, and the increase of the cost of production as well. So we're trying to ease that as best we can. So a little bit contrary to what Arpe said, but the question we're looking to answer is how do orchard floor management practices impact the discrimination of carbon isotope 13 in apple trees? So in this research, I set out to sort of answer three aims. So we wanted to see how the impact of our soil amendments actually changed our isotope discrimination. We wanted to compare that across different phenological stages in the growing cycle. And we also wanted to see if that changed based on growing region, because obviously there are a lot of growing regions in Tassie. So very briefly, what is orchard floor management? Well, it's the practice of managing the soil directly below the trees. The most common that you'll see in Tasmania is a herbicide strip. And the main goal of that is to erase any competition from the root zone for the trees. So that's to maximise your irrigation and to also maximise your fertiliser that you put on. But there are other ways of managing this, and there are other goals as well. So we're also looking to increase the retention of water and reduce evaporation. And we also want to enhance the soil health, and that's particularly important for our cation exchange capacity that makes your fertiliser more efficient. So very briefly, what is water use efficiency? Well, we're looking to change the ratio of carbon that is coming in through the stomatal pores when the stomata is open versus the amount of water that we lose. So if you look at that as a transaction, every time the plant opens its stomata, there's a transaction between carbon dioxide coming in and the water that has to be spent to gain that carbon dioxide. So we want to skew that and make sure that the plant is using its carbon dioxide effectively. And we also want to make sure that we're not losing too much water. So a really good mechanism of actually looking at this is through analysing the carbon isotope composition of our plant tissue. So during photosynthesis, the rubisco enzyme, which occurs in the Calvin cycle, preferentially takes up the lighter carbon-12 isotope over the carbon-13 isotope. So we call this carbon-13 discrimination. And the reason for this is the carbon-13 isotope is 8% heavier, and it also takes about 11% more energy than the carbon-13 isotope. And nature being inherently quite efficient, it's trying to move the lighter and more energy efficient carbon isotope around. So the best thing to take away from this is when the stomata is open or we have low photosynthesis, there's more carbon dioxide entering the leaf mesophyll, and the rubisco enzyme has got plenty to choose from, and it discriminates quite heavily against the carbon-13 isotope. On the other side of the coin, if the stomata is closed or we've got really high photosynthesis, there's a lot of carbon dioxide being pulled out of the mesophyll, and rubisco's got a lot less to choose from, and we end up getting more carbon-13 concentrated in our leaf tissue. So a very quick look at our trial design. We had 3 different regions that we were looking at, Tasmania, New South Wales and WA, and we had 4 different varieties. So in Tasmania, which we'll be focusing on pretty heavily due to time constraints, we had our Southern Bliss treatment, or our Southern Bliss variety, and our Jazz variety. They were all in a randomised block design. We had 5 groups of trees to build 1 replicate, so that's 5 trees in a row, and we tried to keep as little variation as possible between the different sites. But that's not entirely possible to keep it identical. So this just very quickly gives you an idea of what treatments we had and where they were. We tried to keep compost across the board because that's what we were most interested in. As well as that, we had herbicide and our legume grass crop. Our legume grass crop and our grower practice in Southern Bliss, but that's very similar. That just looks like a lot of stuff growing at the base of the trees. So our sampling and analysis, to give us our phenology, we took an early season in November when we had our rapidly expanding fruits, and then a later season again in our mature fruits. So we took leaf and fruit samples there. We then took them back to the lab, we cleaned them, we dried them down, we turned them into a homogeneous powder, and then we took them down to the Central Sciences lab for isotopic analysis. And what that looks like is we superheat them, we strip out everything else, we're left with pure carbon dioxide, and then we shoot that down a hallway through a bit of a bend with a bit of a magnetic field as well. And that discrepancy that you actually get in the trajectory is how we measure those isotopes. So now if we look very quickly at our results, we can see that compost was the most negative. And it's a little bit backwards, but that's indicative of more carbon-13. We also saw significantly higher concentrations in the fruit samples opposed to the leaf samples. And fractionation increased in the later season sampling, except for in our herbicide leaf treatment in Jazz, and that could have just been an anomaly. And as you can see, that very nicely corresponds to our yield, particularly in Jazz. The yield in Southern Bliss, they're very new trees, and it's a very new variety, so they're not quite working the way the grower wants, but we put that down to external factors, not necessarily the treatments at hand. Again, if we look at New South Wales and Western Australia, we're seeing the same trend. So we've got more fractionation in our other treatments opposed to our compost. Compost was the best, therefore, in all regions, and increased fractionation in herbicide and our cover crop / grower treatment. Now, this is where it gets quite interesting. So you can see from the solid line in this figure down the bottom here, that indicates the earlier sampling period. And the dotted lines, those are the later sampling period. Now you can see the R2 or the correlation coefficient actually declines in that later season sampling. And we can also see the other coloured lines, so blue is Tasmania. The other coloured lines are actually moving backwards towards more isotope discrimination. And we put that down to 2 or 3 main causes. So we've got a remobilisation of stored carbohydrates moving from the wood, and they're moving back into our leaves and our fruit, and they're often rich in carbon-12. So that's going to put everything out of whack. We've also got less carbon-13 rich sorbitol coming from the leaves into the fruit, so there's less of that, and that's therefore a less source of carbon-13. We've also got, and the graphs right on the right there will show it better than I can explain it, but our climate is changing from one sampling period to the other, and as you can also see, the coloured lines at the top opposed to the blue, they're very different to Tasmania. So we're seeing a difference in climate, and that's likely having an impact on our vapour pressure deficit and leading to more transpiration, which then again also would likely show more or less carbon-13. So our interpretation and rationale, well, this is where it gets very interesting. So we know that more carbon-13 likely leads to either more photosynthesis or closed stomata. Well, we know that in an irrigated system where all the trees are exposed to plenty of water, it's very unlikely that our stomata have been closed. So we put this down to one thing: compost, and we're going to use compost as an example because compost perform the best in terms of our fractionation. But likely compost has eliminated competition because there's no plants growing underneath. It's enhanced nutrient availability, particularly nitrogen, which is incredibly important for photosynthesis, and it's produced a higher crop load. Now, this crop load, they act as mouths to feed for the plant. There's more mouths to feed, there's more fruit on the tree, you need more carbon. So the photosynthetic demand has increased. And we'll find that when we increase photosynthetic demand, or the assimilation of carbon dioxide, and that's faster than relative stomatal diffusion, that's how quickly the stomata can actually bring in that carbon dioxide into the leaf mesophyll. We find that there is a shortage there, and it forces rubisco to take up the carbon dioxide that it would prefer not to take. So it's a little bit like if you go to your fridge on a Sunday night and you find yourself looking a bit bare, and so you just take what's there. Well, rubisco is a bit like that. It's naturally very efficient. It's looking to take a lighter isotope, but sometimes it doesn't have much choice because photosynthesis is saying, hey, I need some fucking, oops. I need some carbon. So the clean takeaways from this is the compost has a positive effect on our crop load. We've increased our photosynthetic demand, and it's led to less fractionation. So it's a little bit different to the original model that we had in mind, which was we were trying to look at stomata opening and closing, but when we found that they'd been closed, this was our conclusion. The understory cover crops had competed for water and nutrition. It's generally resulted in poorer crop load and decreasing photosynthetic demand. And the herbicide strips, they weren't better, but they weren't worse. So we sort of saw that in our crop load, and we saw that in our C13 composition as well. And quite interestingly, fractionation increased in later season. That makes sense. We've got warmer and drier climates and a higher vapour pressure deficit. But we're also potentially the first study to actually show this using carbon-13 isotopes. So that concludes me. I'd really just like to say a big thank you to Ramandeep and Dougal, who are my supervisors this year. They made this process very smooth, and it was a really enjoyable project to work with. I'd also like to say thank you to Christian down in the Central Sciences Lab for his help with the isotope work. All for statistics, Apple Pear Australia Limited for their funding, the PIPS 4 team for their help with the interstate trials, a bit of project framework, and also sparing me some cash, and Fruit Growers Tas for my scholarship this year. Thank you very much.
Thank you, Rupert. Questions, comments for Rupert? Yes.
So Rupert, that was a very comprehensive presentation. Thank you very much. So you gave us a lot of information in that short amount, but from my understanding of, you suggested that there was increased nutrient efficiency or uptake from the compost.
So increased availability.
Increased availability, thank you. So I'm just wondering, where do you think the source of the increased nutrient availability was, and did the compost itself?
So the hypothesis is, and it's not increased nutrition necessarily, it might be there because the orchard is fertigated, but it's more or less providing more available cation exchange points in the compost. So you've got a lot of organic matter down there. It makes your soil more sticky, so to speak. And so you've got more binding sites for that, for those nutrients to actually stick around. And they also might be more bioavailable. You've also got more microbes down there, so you might be mineralising a bit more of your nutrition out of the soil, opposed to, and I know definitely down in Ranelagh where it's quite a sandy soil, the nutrition that they are putting on, it doesn't last very long. And a lot of it becomes unavailable because of the acidity of the soil profile.
So you feel that the compost is not adding extra nutrients, because that's what people put compost on for, right?
Yeah, I mean, it would have nutrients in it, but I think the main point is it's maybe holding those nutrients for a bit longer.
Yeah. Thank you very much. Thanks.
There's a question online
Yeah.
for you, Rupert. Actually, this one for Sarah.
Oh, really?
Yeah, but yours says, do you have carbon nitrogen ratio soil data to back up the claim that nitrogen was more available?
Yeah, so we don't have data in the soil, but what we did take, and it's good for me because this is such a part of a much bigger project, we do have nitrogen data in the leaves. Now, I didn't include it in this because I already had enough to talk about, but we did see a correlation between that nitrogen in plant matter, particularly in the leaves and the fruit, which matched with the compost treatment performing better.
Back to the room. Any other question?
Probably a dumb question because I didn't connect something. At the first slide, you said climate change, water costs and production costs.
Yes.
Can you connect the dots for me, please? From this work, how does
Yeah,
it
absolutely.
relate back to that?
Well, if we take a step back and we look at the original thought process before we started talking about photosynthesis and things like that as well, the original aim of the game was to make our orchard floor less vulnerable to changes in climate. So you have a long spell between rain, potentially that compost is buffering it by retaining a bit more soil moisture. You know, the dam's a bit empty. We can't irrigate today. Ideally, that's not what's happening in an apple orchard. But ideally, we can buffer that, prevent a bit of evaporation from occurring. And we can also stop the other grasses and weeds, which there's plenty of in that orchard, from actually taking up those resources that we want to put into our plants, into our trees.
And your findings, to what extent do they
Well, I think if we look at compost, you know, we got a much higher yield, particularly in jazz. And we saw that photosynthesis increased, so its capacity, which means that the carbon dioxide that's actually coming through the stomata, it's going into that into the cellular airspace and into the mesophyll, is actually getting utilised, opposed to they're opening the stomata, they're bringing in a heap of carbon dioxide, but I'm not going to use all of it because I don't like it, it's too heavy. We're actually forcing everything to use it from that drive. So it just means that, let's imagine you're stacking crates and you're picking up two, but you're only selling one of them.
I got a little bit stuck on the carbon-13 story.
Yeah.
So initially, it sounded like you were saying the plant's going to have more uptake of carbon-13 if its stomata are shut.
Yes.
And it's low, it hasn't got much moisture. But then at the end, you're saying the opposite of that. So how is carbon-13 discrimination a good measure?
So do you mean the values? Because the values are recorded negatively, which makes it a bit confusing.
Right, maybe that's where I'm getting lost, because it seemed like at the end you were saying.
Yeah, so carbon-13 it's measured relative to the atmosphere. So the atmosphere sits at what they call, it's a very small number, so they talk in thousandth instead of percentage of 100. And it's recorded as a negative of relative abundancy. So in the atmosphere, it's sitting at 6.6 and it's marked as a negative. And then when we're looking at it, to keep that trend, when we're looking at it in the plant, we continue to mark it as a negative. And actually a decrease in that composition is the further away it gets from that -6. So we get down to -29, for example, that's further away, there's less of the carbon isotope.
That means that the plant has taken up less carbon-13 than is available.
It does, yes.
There's one more question online for you.
Yeah.
So has the CEC also been measured? If not, what would this be?
Yeah,
What
so
would be the next
it is part of a much bigger project. and I've only got so much time in an honours year, but it is also a continuing project. I did have a slide in there that said what we would like to look at in the future. Probably ate a bit too much of my time, but I think moving forward with this project, looking at the actual soil properties would be the next best step, and also correlating that back to other dynamics in the tree as well, just to really confirm what we think's going on.
You go, Mike.
Oh, no.
I was just about to say, and there's probably a fruit quality implication all this as well. Without going into details there, how likely would you be to recommend composting as a strategy for managing orchard flora for growers, given its high cost?
I would recommend it, but we would need a way of putting it on efficiently. So we filled a 12-yarder truck just to meet our 5 replicates in each block. So that's sort of like 10 tonnes of compost went on. That's all well and good because you can apply it and it sticks around for a very long time, but we need a way of applying it well. So whether it's a converted feed trailer that's actually shooting it down and it's not Ramandeep shoveling it, because I don't think Ramandeep wants to travel around Tassie and shovel compost.
When I was at school, I remember learning that the carbon dioxide concentration in the air was 330 parts per million. And I think more recently it's around about 450 or possibly even more. Has that affected the yield of apples and other plant material, as far as you know?
It's not something that I've looked into.
Yeah.
It would be very interesting to know. You can imagine, based on the physiology of abundant carbon,
Sure.
and the efficiency of bringing carbon in, well, obviously that carbon is diluted in other air particles, and the stomata has to manage all of those air particles in front of the stomata in the boundary layer before it can actually bring those through. So you'd imagine if it's more easy to grab and there's more of it in the airspace and more of it in the boundary layer, then yes, it probably would, because at the end of the day, every time they're opening that stomata, and this is the trees, they can get the carbon dioxide they need and they lose less water in the process.
What about the medicinal cannabis glasshouse, Rupert?
Yeah, well, they're increasing carbon content.
Carbon dioxide for exactly that reason. So they have a carbon dioxide store and they aim for 1,000 parts per million.
Right.
Because it increases productivity for exactly that reason.
I do have another question, but it's got to be the last question.
Yeah.
I'd be very keen to hear what you think the value of doing an honours year is in terms of your employability and how it's setting you up for life.
If you're not afraid of hard work, I would recommend it to anybody. Going through my undergraduate, I thought, oh, it's just another year. It's just another year. And then you think about it, it's not just another year, it's 33% more of a degree. And it's 33% more of a degree that's really it's a bit of a melting pot, really. You combine all of the knowledge that you've picked up over the previous 3 years and you have to apply them in a slightly more strenuous task. You've got to present it in front of people, and you're working on a much bigger project on a very specific topic for a lot longer period of time. So you become a bit of an expert in a microfield of sorts, opposed to in your undergrad, you're doing lots of little experiments. Only 20% of them actually work. And you do very what I like to call shallow learning. So you only write what you know is going to appease the person marking it, opposed to the most interesting facts. Whereas honours, I've got a bit of freedom to actually talk about what I want to talk about. And the marking is going to be irrespective because the markers don't necessarily actually have expectations of what they're going to be reading.
And I guess that just wasn't the question just for Rupert, but Sarah as well.
You repeat the question.
Oh, the question was what is the value of doing an honours year?
I think for me, I've learned a lot about how to manage myself and my time and manage my supervisors. Because I did have 4 supervisors by the end of it, which can be very hard to coordinate meetings. And also I did an ethics approval, which I don't wish on anyone. Yeah, I feel like I learned a lot of hard things which will serve me well going forward in management, especially. And also just how big the world is, like going to Fiji was amazing.
And I think just to add to that, I think at the end of last year, I thought I knew it all. And then I step into the end of this year and it's like, wow, I know a lot more. So I think the learning experience and the learning curve is a lot steeper in honours. It really forces you to bring in a lot of complex information, and whilst it feels like it doesn't really stick and you read a lot of different reports, right at the end of it, all of a sudden you get asked some questions and you can answer them. So it's really nice.
Thanks.
Thank you.
Good afternoon, everyone, and thanks for joining us this afternoon for this last and final honour seminars for this year.
Thanks, Alpay. So my research project focused on the capacity of existing soil rhizobia to buffer against the a commercial inoculant in Tasmanian soils. So legumes are valuable fodder crops in Tasmania, and they're significant biological nitrogen fixers due to their association with rhizobium bacteria. It's a naturally occurring symbiotic relationship, so it's really important for producers to optimise its impact on production, especially for low input extensive systems that don't have as much access to synthetic fertilisers due to such a large scale of production. So we already know how rhizobia infect legumes. So legumes will emit flavonoids, and that encourages rhizobia to emit nod factors, which signal to the root hairs to generate an infection thread. So you can see in this diagram, the rhizobia will enter through the infection thread and they'll go through the cortex and into the space known as the nodule primordium. And from here, the nodule will develop and bacteria will turn into bacteroids. And with nitrogenase, they convert nitrogen gas into ammonia, which is transferred throughout the plant. And as you can see up the top here, you can see an actively fixing nodule due to its pink colour. And this is due to the presence of the hemoglobin, which creates an anaerobic environment for the enzyme to do its job properly. We also know that there's several limitations to nitrogen fixation, including saline soils, soil moisture, organic content, and nitrate levels in the soil. And we also have had studies that found that inoculants are strong nitrogen fixers initially. However, this often failed to persist long term. This is largely due to either environmental conditions or existing strains that have better adaptations to local soil conditions, and they outcompete inoculants and recolonise roots. There's also been studies done on horizontal gene transfer, which incurs native existing strains to transfer genes to inoculant strains. And in doing so, inoculant genetics are really unknown, and fixation limitations makes it hard to determine fixation, efficacy, and their impact on production for producers. So the problem, so there's a large knowledge gap in the long-term persistence of inoculants in Tasmanian soils. And due to a heavy reliance on pre-inoculated seed, farmers really don't have a lot of options sometimes, and they have to purchase pre-inoculated seed, and they really don't know how old it is or the quality of it, and a lot of the time, due to no fault of their own, they'll store it in sheds that are too hot, and it really just reduces the efficiency of the rhizobia at sowing. And yeah, this really has long-term impacts for perennial legume and rhizobia relationships. So my aim throughout my project was to investigate the persistence of rhizobia in pasture legumes in Tasmanian farming soils, and I had specific aims related to each of my aspects of my project. Number one was to investigate the productivity and nodulation performance of an 8-year-old legume plot. and to investigate if existing rhizobial strains in Tasmanian soils can compensate for commercial inoculant failure. So one aspect of my project was field sampling at the Cressy Research Station. It consisted of an 8-year-old pasture with numerous species and cultivars. It included 64 research plots, and in each plot I collected 2 plants. And this was to assess for nodule score, nodule size, plant root length, and to observe persistence. And this is just an example of my nodule scoring chart that I use. It's a standard MLA nodule scoring chart, and it provided a way to quantitatively measure nodulation across the species. So it starts at 0, so there was no nodules, and it goes right up until 8, extremely abundant. And it just looked at colour of nodules, number of nodules, size of nodules, and distribution of the root system. So at the site, we observed persistence. We observed poor and good persistence. So poor persistence included that there was no white clover observed at all over that at the 8-year period. And there was limited Caucasian clover and few red clover present. And overall, it was just patchy growth, low density regrowth in a lot of the species. And this is likely due to the extended drought periods and rainfall conditions that have been experienced throughout those 8 years. We did see some good persistence, but it was largely due to root physiology and spreading habits. So lucerne was a long-lived plant, and many of the ones that we sampled were original plants from sowing. Whereas strawberry clover, semi-dormant, which means it was able to be a bit more active before some of the other species, and it was able to be spreading and popping down new plants a lot more efficiently than some of the other species. So part of my key findings were the Cressy soil data. So the Cressy soil had a slightly acidic soil of 6.08 in water and 5.46 in calcium dichloride. And the organic carbon content was at 1.87%, which was sort of low to moderate for rhizobial function. However, long term, this could impact rhizobial function long term. Nitrate levels were at 39.1 parts per million, which is sort of moderate for pasture legumes. This could have suggested that nodulation was suppressed due to the availability of nitrogen in the soil. We also performed PCR to determine the presence of the nitrogen fixing gene, and Cressy came back with a strong positive result, which is indicated by this red arrow. So key findings at Cressy field data, we focused on between species. So strawberry clover scored the highest in nodule score and size. You can see it's between 4 and 5 on the nodule score, which is adequate and ample. Lucerne was really limited in its nodule size and score, well below 1 in the nodule score. And overall, all the nodules were really small, below 3 millimeters across all species. Even though strawberry clover received the highest results, it's still at average. So in terms of optimal production, it's still quite average. But whether or not this is good for a long-term pasture, it's still doing relatively well. We also took strawberry clover and assessed it from its numerous cultivars and breeding lines that were present at the site. So as I said, nodule score remained between 4 and 5, so that was fairly average up to ample. Nodule size was still small, under 3 millimeters, and this is all likely due to environmental factors that limited nodulation efficacy or the presence of existing rhizobial infection. This is an example of the different root physiology of the plants at the site. So you can see here, white clover had really shallow root systems, which is probably a reason for it no longer existing at the site. It wasn't able to be resilient through those drought periods. And you can see lucerne has a really extensive taproot. It was able to access a lot more moisture over this period. However, in that 8 horizon, there was really limited nodulation, which is usually expect nodulation to be towards the crown of the plant. The other clovers, they all had their own ways of spreading and putting down new plants. So red clover was able to spread by seed, and strawberry clover via stolons, and the remaining via rhizomes. So they were able to put down new plants every season and they were able to recolonize root systems. So it's likely that existing strains had recolonized these plants. So my field study conclusions was that nodulation was low to adequate across species, indicating that limited early season rhizobial activity. So we sampled in late winter in about August. So it's really traditionally a period of low growth. So if we'd sampled in spring, we might have seen some different results in a period of high growth and water access. And plant physiology and spreading habits really influenced persistence of the plants themselves and also provided opportunity for existing rhizobia to infect root systems. Strawberry clover showed that highest nodule score in size and that long-term plant persistence. And overall, my research showed how important it is for low input extensive systems to really optimize rhizobial legume relationships to ensure that nitrogen supply and reduce the reliance on synthetic fertilizers. I also had another aspect to my project, which was a pot trial. It included 4 soil textures, 4 pasture legume species, then non-inoculated and inoculated treatment. The main aim for this pot trial was to understand how each soil was able to nodulate the plants without being inoculated. And yeah, that just provided a way of understanding how the soil responded. Future research, I'd like to further analyse the results from my pot trial. I think it would be really beneficial to understand long-term by setting up trials to assess nodule development and fixation, assessing nodules at 2, 4 and 6 to 8 year development. And during these time periods, isolating the specific rhizobia strains to see if they are the inoculant strains over time or if existing strains have taken over. I'd like to extend many thanks to my supervisors, Dr. Shannon Fall and Dr. Ron Smith, as well as Honours Coordinator Dr. Arpe and Caroline Clay and Zach for their lab tech expertise, Phil Andrews for his help with my greenhouse trial, and everyone who has supported my research thus far.
Right, thank you very much, Shannon, for your presentation. I'd like to throw the room open for questions. Probably Shane will help us coordinate the room because I might not be able to see any hand that's raised there properly while I look for hands that are raised online here.
Anybody? Rowan has a question.
Really good presentation. On the sampling at Cressy, so that trial was sown, was that inoculated when it was sown?
Yeah, definitely, yeah.
And then were you able to distinguish whether or not it had been colonised by a wild type inoculant or what's that? There's some PCR stuff there.
Yeah, no, we weren't able to do that. We did plan to do that at the start of my research project. We sort of had to adapt because we weren't able to isolate the strains. But that's sort of why I had my pot trial set up, so I could have those. Because we included the same soil that was from that site. And yeah, we wanted to compare one that was inoculated and one that wasn't and see what would happen.
Cool. And those results from the pot trial still count then?
Yes, we're still analysing them to work out what's statistically significant and what else, but yeah.
Oh, very good.
Is there anyone popped anything in the chat, Archives?
None at this stage yet.
Anyone else? Alita? I'll try and think of a question. Thanks for the presentation, Shannon. That's good, really clear. So at Cressy, I wasn't sure the background, but you say it's low input.
Yes.
Does that mean they're actually not applying any fertilizer at all?
I would not know. I doubt that they are. I wouldn't really.
I don't reckon it would have been hit with any nitrogen.
No.
Okay. So it's a very low input.
It's sort of been left there to persist for those period of times to see how it behaves.
And it's being managed by sheep or dairy? What's feeding on it?
I don't think anything is. I think they've put some sheep over it a couple times. Well, I'm not sure how many times, but I know they've put some sheep over there. But
well, you could think that if it's a system with animals, that they may influence the survival of the plover, since they may be preferred compared to pasture.
Yeah.
So it might be something to consider in that history.
Yeah.
Yeah.
Yep.
Yeah, basically that's just a comment. Yeah. All right, thank you.
No, you're right.
Anyone online?
Not yet. Just that Roman added a comment that grazed sheep occasionally lead to low fertilizer input.
Sorry, what was that?
Say that again. We can't see the chat
Oh,
comments.
he put a comment in the chat saying that grazed by sheep occasionally. I think talking about the crazy
Oh, okay. Confirming it, yeah.
and so has low fertilizer input.
Yep.
Yeah.
So what with low fertilizer input then, Shannon, what do you expect that, what effect do you expect that might have had?
Yeah, well, the higher the nitrate levels are, traditionally the more suppressed nodulation would be, because the plant's not having to, the plant's got all the nitrogen it needs, so it doesn't have to have that relationship. Whereas the nodulation doesn't have to be as efficient because it's got that there available.
So why do you think you had, like you said, they were low to average scores?
I think environment has a lot to do with it. Because of the low periods of rainfall, and there was a couple of extended drought periods. Plus, before I sampled, there was the site didn't get an autumn break, so there really wasn't a lot of rainfall in autumn to get that roots. Yeah, the roots activated, and so yeah, I think overall the roots and the growth of the plant really limited. I think we might have seen a bit different results if it was springtime and the roots are actively growing. We might have seen more nodulation.
It'd be wrong of me to ask difficult questions, so.
Any more question online? Anyone online? No.
Well, we can finish with Mike's question that he asked both the other students the other week about what do you think you got out of doing your honours year as compared to if you just graduated last year?
Yeah, I think it's definitely made me think a lot more about how I do things. Definitely made me got to be a lot more organised for sure in my honours year. Planning, pretty much everything about running an honours project makes you a lot more organised and planned. And I think it's helped me that way. And obviously learning more about a specific topic. Yeah.
Thank you. Pass that on.
All right. Thank you very much, Shannon, for your responses to the questions, and thanks everyone for making it happen by joining us again online this very afternoon. I would ask that you help join me also to thank Shannon